No, sir.
Okay then. So we will now discuss about two techniques today. These are immunological techniques. One is ELISA and another is RIA.
Yes,
So
sir.
I hope you might have heard about these techniques earlier. What is
Yes, sir.
ELISA?
Sir, ELISA is enzyme-linked immunosorbent assay, and with
Okay.
the help of it, we detect the antigens or antibodies in the sample.
Okay. Then why it is called immunosorbent? Yes, Samra, can you see my slides?
Yes, sir.
Okay. So why it is called immunosorbent? Yes, can you hear me, Samra?
Yes, sir.
Okay. So you do not know the answer?
No, sir.
Okay. And why it is called, what do you mean by enzyme linked?
Sir, because the antigens or antibodies are detected by adding the enzyme, which changes the color for certain substrates. The
Okay, okay.
enzyme.
So you just try to understand it. When we use the word immunosorbent means an immune molecule is adsorbed. Sorbent word came from absorption.
Absorbed. Okay, sir.
Absorbed, adsorbent. So the adsorbent here is a immune molecule, which is either an antigen or an antibody.
Antibody.
I hope you are very much clear about the word antigen and antibody. What is an antigen?
Yes, sir. Sir, antigen is any foreign substance that is introduced in
Hmm.
the body, and
Okay.
antibodies are produced in response of the antigens.
Okay. So if we are introducing alcohol in somebody's body, will it generate immune response?
No, sir.
Why not?
Sir,
So what is an
antigens are
antigen?
generally protein molecules, protein substances.
Okay, it may be protein also, it may be peptidoglycans also, it may
Okay, sir.
be like glucose linked molecule also. But yes, it should have a particular size.
Size.
If the
Yes, sir.
size of the molecule is a particular size, then only it will be having antigenic property. And due to
Yes, sir.
that antigenic property, it will elicit the immune response in animal body. And
Yes, sir.
then against that antigen, the animal body will generate antibodies. Okay, so we
Yes,
have
sir.
two immune molecules. One is antigen, and another is antibody. Right. So
Antibody.
we want to measure the concentration of these immune molecules. So what is the like use of this measurement? How it will help? Yes, Samra. What is the use of this measurement? Why we are measuring it?
So we are measuring the antigens or the antibodies in order to detect that if there are certain disease present or not.
Okay. Okay. So it is largely like assessment of infection.
Yes, sir.
Right. So generally, when we talk about like antigens, the antigens are mostly foreign protein or like peptidoglycans, which are introduced inside the body. So mostly these are say bacterial cell wall or bacterial secretory proteins or fungal cell wall or fungal secretory protein, or we have the viral coat proteins or the like protein secreted by viruses. Okay.
Okay, sir.
So all these molecules, they when they enter inside the body, they elicit immune response. So if we simply measure the either antigen or antibody in the fluid, largely it is blood. So we can just assess that what is the status of infection,
Yes,
which
sir.
infection we have. So these are the techniques of molecular diagnosis. These are the technique of molecular
Molecular
diagnosis.
diagnosis.
Generally, we say na, that diagnosis is done on the basis of symptoms.
Yes,
So
sir.
largely we have symptomatic diagnosis, but sometimes symptomatic diagnosis may be misleading. So to avoid that, we generally go for molecular diagnosis. Okay.
Okay, sir.
We go for molecular diagnosis. So in molecular diagnosis, we measure the antigen or antibody molecule, and if we are able to measure a specific antigen or a specific antibody, then we can be sure that the symptoms are because of this particular
Particular
infection.
antigen.
Okay.
Okay, sir. Okay, sir.
So how it is done? When I said immunosorbent means that any of these antigenic molecule, antigen or antibody, it is adsorbed inside a plate, a
Okay, sir.
polystyrene plate. So since it is adsorbed, it is on the surface of the plate. That's why we call it immunosorbent, right?
Yes, sir.
And that immune molecule is linked with the enzyme, which work as a reporter enzyme. How
Okay,
do
sir.
we know that whether the molecule is present or not, and how much quantity it is present? Then we use link it with the enzyme. That's why the technique is called enzyme linked immunosorbent.
Immunosorbent assay.
Okay.
Okay, sir.
So the technique is like how it goes, that this thing like what we were discussing, like antigen antibody of interest, it adsorbed on a plastic surface. That's why it is called sorbent. Antigen is recognized by specific antibody. So it is recognition is by with like antigen antibody reaction. That's why it is a immunosorbent, because it is based on a immunological reaction. Now the antibody is recognized by a secondary antibody which has a enzyme attached. That's why it is called enzyme linked. Okay. Now the
Okay,
substrate
sir.
of this enzyme is like poured in the well, and then we can find out there will be there will be reaction which generally leads to color generation. Okay, generally
Okay,
leads
sir.
to color
Color
generation.
generation.
In ELISA we have color generation. In RIA we have radioactivity. Okay, so and we measure that
Okay, sir.
color. Now the intensity of the color will decide about the
Amount of.
amount of the antigen or antibody present, and the
Yes, sir.
amount of antigen antibody will be directly proportional to the load of bacteria,
In.
fungus, or virus in the
Yes, sir.
body. Okay, so
Yes,
higher
sir.
the color,
Okay, sir.
higher the load, and we can find out
Mm-hmm.
that what is actually the status of the infection. Okay,
Infection.
so this
Yes,
is
sir.
history about ELISA, who developed it. So it was basically developed by Rosalind Sussman Yellow and Solomon Bresson. They published first paper on it was some immunoassay they proposed, and later on 1961, like Peter Perlman and Eva Engevall at Stockholm University in Sweden and Netherlands, they independently developed the papers regarding the development of ELISA.
ELISA.
Okay, so the basic terms are like we have a solid surface which is working as an adsorbent surface, and generally you'll find that we have a 96 well plate which is having say 12 column or 8 rows, whatever you want, whatever way it comes.
Okay, sir.
So it is I think you might have seen it, but it is one of the most commonly used ELISA plates. Okay,
Yes,
then
sir.
simply first we do that. In first process, the antigen or antibody is adsorbed on the surface of the solid
Surface.
surface.
Yes,
And
sir.
generally, like in the lab, we do not do this procedure, because generally we get a pre-adsorbed plate. So suppose if I want
Mm-hmm.
to test for say TB, then I will get the pre-adsorbed plate. If I want to test for HIV, I will get the pre-adsorbed plate with the
Okay,
antigen
sir.
or antibody based on the
Antibody.
activity I
Okay,
want
sir.
to perform. Right? Now,
Okay, sir.
what is done actually? Like what happens that like first we do the washing of the plate, so that if there is any unbound antigen or antibody present in the well, it may be removed.
Okay,
Then
sir.
then what we have, the common enzyme conjugates that we have, like antigen antibody, I already explained. So the
Yes,
common
sir.
enzyme conjugate is horseradish peroxidase. Horseradish peroxidase. It's
Peroxidase.
a common enzyme, uh, like common enzyme that is used for assay. Then chromogen is a chemical material which work as a substrate, right, for that enzyme. And as a result of this addition of this substrate, there will be a colored reaction, and that colored reaction will be used for assessment of the quantity, right? Then the
Right, sir.
reaction can be stopped by addition of another like an enzyme of an substrate. Then the reading can be taken in a spectrophotometer. So these are the basic step of the
ELISA.
uh, ELISA. And principle wise, it is a lock and key system. Means we have we explore the immune reaction between antigen and antibody. Okay.
Antibodies. Okay, sir. Okay, sir.
In equipment, we require a 96 well plate where we have to perform the assay.
Assay.
Then we require multi-channel pipette. Generally, a 8-channel pipette is used so that we can do titrations, because titrations are very crucial in ELISA. Then we have a washing device, because every time we have to wash it so that the unbound conjugates can be removed, right?
Right, sir.
And then we have the, like, these are the microplate washers which are available in the market. And then finally we have to read it. Finally we
Mm.
have to
Read
read
it.
it. So this is a reader, eh? So
Okay,
this
sir.
can be read by a uh, spectrophotometer, right?
Okay, sir.
So in general, what happens, like you just see, this is the well of a microtiter plate, and you see you have an antigen adsorbed on that.
Yes, sir.
Now, if you put a body fluid, now the body fluid may be blood, body fluid may be something else. Huh? It may be
Yes, sir.
cerebrospinal fluid or whatever. Now, when you will put a body fluid inside this well, then if the body fluid is having antibodies specific to that antigen, those antibodies will bind, right? Now, these antibodies are
Yes, sir.
specific, like say IgG antibody or IgE antibody or with whatever antibodies we are like considering. Now, against that antibody, we have already generated a secondary antibody. Now, how these secondary antibodies have been generated, from where we get the secondary antibody? Yes, Samra, from where we get the secondary antibody? Yes, Samra, from where we get the secondary antibody?
See, sir, we can synthesize it or we can purchase it.
Synthesizing and no, we can purchase it, but how it is
Yes, sir.
generated? How it is generated? Synthesizing antibodies
We
is
can purchase it.
impossible. We
Yes, sir.
cannot synthesize
Hmm.
it. We have to
We can
purchase it,
purchase.
but how it
Yes,
is?
sir.
But how it is generated? What do you mean by secondary antibody, and how it is generated? Yes, Samra, can you hear
Sir,
me?
I don't know.
Actually, what happens? Suppose there is reactivity of an enzyme with human IgG. Then that human IgG is isolated first, and then that human IgG is injected in some other animal, right? Either in,
Okay, sir.
say, rabbit or goat. Mostly, like we do it in rabbit or goat. Now,
Okay, sir.
rabbit and goat, human IgG is what?
Antigen.
It behave like an antigen.
Mm-hmm.
So the rabbit or goat, it will generate antibody against human antibodies, right?
Okay,
Because
sir.
for
Okay, sir.
rabbit or goat, that human antibody is not an antibody, it is an antigen.
Mm-hmm. Yes, sir.
So these antibodies which are generated in some other animal, they are
Mm-hmm.
known as
Secondary antibodies.
secondary antibodies, right? So mostly
Yes,
these
sir.
antibodies, either these antibodies can be generated, earlier it was through animal models largely. We used to generate in rabbit or goat. But nowadays we have also started following the concept of like monoclonal antibody formation through cell culture systems. Okay,
Yes, sir. Yes, sir.
that we have a more purified and more coherent product, and
Yes, sir.
isolation of those antibody from the, say, goat blood or rabbit blood is again a tedious task, so we can save
Mm-hmm.
ourselves from that tedious task. Okay. Now
Okay, sir.
this secondary antibody is linked with horseradish peroxidase enzyme,
Mm-hmm.
because this we have to buy from market according to our requirement. Suppose this antibody I suspect it is IgE, so I will buy IgE specific antibody. If this is
Okay, sir.
IgG, I will buy IgG specific secondary antibody. Okay, so
Antibody.
whatever.
Okay,
Yeah,
sir.
so
Okay, sir.
yeah, okay. So
Okay,
these
sir.
antibodies are purchased from market, and then these are always come conjugated with a enzyme called horseradish
Enzyme
peroxidase.
peroxidase.
Now, in this well, what happens? Like if you see the procedure, first we will pour the human blood. So if it is having that antibody, like if the person is having infection of that organism which is having this antigen, then this antibody will be present in the human blood. So antibody will bind to this antigen. But there will be so
Okay, sir.
many antibodies which will be there, which are
Mm-hmm.
unbound for. So then we do washing of this well, so that
Mm-hmm.
all unbound antibodies are removed. So we only have the bound antibody, right?
Yes, sir.
Then in this well, again we put the secondary antibody which is linked with the enzyme. And then again we have to do washing. Why we have to do washing here? Why we have to do washing here? We have to do washing here
Sir.
because there may be so many
Mm-hmm.
secondary antibodies also which
Mm-hmm.
are unbound. So
Mm-hmm.
all these unbound antibodies are removed. So now we have primary antibody bound to the antigen,
Mm-hmm.
secondary antibody bound to the primary
Primary
antibody,
antibody.
secondary antibody is having enzyme
The enzyme.
horseradish peroxidase linked to it. Now we will put the substrate of this horseradish peroxidase, and then this like one enzyme substrate enzyme reaction will take place, and we'll get a colored product.
Correct.
Now
Okay, sir.
this plate can be read through a plate reader, and the plate reader will tell that what is the intensity of the color, and based on that we can identify that whether there is a some infection is there or not. Okay, so this is basic
Okay, sir.
idea of the ELISA. Now ELISA are largely we say these are three types: non-competitive ELISA, competitive ELISA, and multiple or portable type of ELISA. We'll discuss. Huh? Okay.
Okay, sir.
Now, non-competitive ELISA are direct, indirect, or sandwich. So largely we perform non-competitive ELISA. Mostly we perform non-competitive ELISA. Okay.
Okay, sir.
So in direct ELISA, actually, what is there in non-competitive direct? It uses a primary labeled antibody that reacts with the antigen. Okay. It can
Yes,
be
sir.
performed with the antigen that is directly immobilized in the assay plate. Not widely used because there are certain issues. Like what happens in this ELISA, I think I'll have this one. You can see here. In direct, what we have, you just understand from this graph. So what we have, the primary antibody itself is having the enzyme, right?
Okay, sir. Okay, sir.
Like suppose I know that if this infection is there, then generally suppose IgG response is given. So nowadays we have a facility that we can send, like using cell culture based methods, we can isolate like a lot of like a human IgG can be produced. Okay, human
Yes, sir.
IgG can be isolated also from human blood, but that is again a tedious task. So instead of doing that, we can produce
Hmm.
monoclonal antibodies from the hybridoma
But
technology using
yes, sir.
hybridoma technology. Okay,
Yes,
and
sir.
those antibodies are then linked with the enzyme. So this
Hmm.
is direct, means this antigen, its own antibody, and then antibody is
But
tagged
it's tagged enzyme.
with enzyme. So directly you put this like you take a plate which is having antigen adsorbed in that. That is a pre-coated plate available in the market. You simply put the labeled antibody, and then you will get the result. Okay.
Yes, sir.
In the second case, but these are like not very sensitive. Okay,
Okay, sir.
not very sensitive. Now you take here in this case, the one I told you, it's an indirect one. So we have the antigen on the plate surface, then we have a primary antibody, and then we have a secondary antibody.
Secondary. Okay,
So
sir.
this is called indirect, means we are measuring the concentration of this antibody, but by linking it with another antibody, right? You
Right,
got
sir.
it? So this way
Yes, sir.
we can generate it. So we have direct and indirect. Then we have sandwich ELISA. It is one of the most commonly used ELISA Because what happens in these type of ELISAs, suppose this antigen is having say one epitope, and this antibody recognizes this epitope and binds. Okay,
Mm.
that
Yes, sir.
means like there may be a false positive reaction, because if it binds, it will give a color, right?
Mm-hmm.
So that
Yes, sir.
reaction may be false positive. So to enhance sensitivity, we got a concept of
Sandwich ELISA.
sandwich ELISA. Now this is a picture of sandwich ELISA. You understand? I'll try to understand that instead of coating a antigen on the plate, what we coat
Antibodies.
what we absorb antibodies.
Yes, sir.
Then the target protein or the target molecule is an antigen which is present in the where?
Sample.
In the sample, say human blood.
Mm.
Like if
Yes, sir.
somebody is having infection, then we are, so here the difference is we are measuring what? We are measuring
Antigens.
antigen. We are not measuring antibody, we are
Mm.
measuring antigen. Now
Antigens.
advantage with this one is that like this you see that this antigen is conjugated by two antibodies. That means it becomes highly specific.
Highly sensitive. Yes, sir.
Like if first one, like this antigen has to bind with its capture antibody, this antibody is called capture antibody. If it doesn't bind, then no reaction will take place.
Yes,
Then
sir.
you have a detection antibody which we can call a like there it was called primary antibody, but here it is called detection
Detection
antibody.
antibody.
So this antibody will again bind to the antigen. Now you see that the antigen is bound by two antibodies. So if this reaction doesn't takes place, this binding doesn't take place, no reaction, negative. If this reaction also doesn't take place, no reaction, negative, right?
Yes, sir.
That means the antigen is sandwiched between two antibodies. So
Yes, sir.
this enhances the sensitivity of the assay. Okay,
Okay, sir.
this enhances the sensitivity of the assay. Okay.
Okay, sir.
Then we have again our secondary antibody, which is linked with the HRP, and then it gives a color. So this technique is known as sandwich
Sandwich
ELISA.
ELISA.
So why we call it sandwich? That the antigen is sandwiched between two
Two
antibodies,
antibodies. Yes, sir.
and this is one of the most you know like very sensitive technique
Yes, sir.
which we are using. Okay. Now
Okay, sir.
competitive ELISA. This also we perform. Now, how competitive ELISA is performed, you just try to understand from this picture. So, what happens actually that you see, like this, these are indirect ELISA that we discussed. Like we have the antigen, then we have the primary antibody, we have the secondary antibody, and then we get the color. Okay,
Yes, sir.
okay, you are getting it.
Yes, sir.
Then we have sandwich where antigen antibody is bound. We have an antigen, we have a detection antibody, and then we have a
And then.
secondary antibody.
Hmm.
And again, we get the color. Okay.
Okay,
Now
sir.
in competitive, what happens that like this and indirect is generally used for detection of HIV,
Mm. It's
and sandwich is used as a tumor
tumor
marker for
markers.
detection of hormones,
Hormones.
and competitive is used for detection of free testosterone. So
Testosterone.
this is also used. So what happens in this one? Like you have antigen bound on the surface, right?
Okay,
So
sir.
you incubate the antigen, the antibody with the antigen to be measured. Okay. So
Okay, sir.
you like in a well. So you have the antigen antibody reaction, and some of the antibody will bind to the well. Now
Okay, sir.
what happens that we also give unbound antibody. Now this is bound antibody and this is unbound antibody.
Unbound.
So if we give this unbound antibody, now this unbound antibody will knock out certain antibody which is already bound, right?
Okay.
That means the
Mm-hmm.
antibody is competing with the antigen, and one of the antibody is linked with the like if some of the bound antibodies is kicked off. So that means there is a competition, because whether
Mm-hmm.
the antibody is having a reporter molecule or it is not having a reporter molecule, that doesn't affect its binding, right? So a
Yes, sir.
molecule, a antibody with reporter molecule or antibody without reporter molecule, that means they both are competing for its antigen.
Antigen.
Okay. So these like these three techniques are widely used. Then we have multiple and portable ELISA. These are basically newer technique that uses solid phase mode up on an immunosorbent polystyrene rod with 8 to 12 producing. And then how it
Projects.
happens that the like this ogive pins, actually these are bit advanced technique which have been introduced very recently. So instead of having a like plate kind of structure, we have these ogive pins and racks, so that just you dip and you will get the result.
Okay, sir.
And then we have the like modified ELISA, where we can have like the biotin avidin complex, like you have the like monoclonal antibody coated on the well, then we have a capture antibody, then antigen is poured in that, antigen will bind that antibody, and then we have a like antibody which is conjugated with biotin.
Biotin.
Then we
Yes, sir.
have another molecule called streptovidin. It will bind to biotin, and that streptovidin is having conjugated
HR.
with HR. Okay.
Okay, sir.
So this way, just to enhance the sensitivity of the technique, it has been uh, done. Okay.
Okay, sir. Okay, sir.
Now, this slide is about the measurement of plates, because this is very important aspect of any ELISA, that how the plate is measured. So we use plate readers for that. Okay. So
Okay,
we
sir.
can measure an absorbance based on like mostly HRP is measured at 450, like substrate. It gives a color which is measured at 450 nanometer. Okay.
Okay, sir.
So now you see here, like if you get color, it is positive. If you do not get color, it is negative.
Negative.
So for patient A, you can see it is positive.
Positive.
For patient B, for patient C, like this you can see.
Okay, sir.
Like maybe for patient A, it is negative.
Negative.
Patient is having infection, but it is low.
Mild
But
infection.
patient
Yes,
C,
sir.
you can see very high infection.
Yes, sir.
Okay. So that way
Okay, sir.
we do it. So for in every plate, we need to have a positive control. We need to have a negative control. Positive control means we'll have the highest color.
Mm-hmm.
Negative control means there will be no color.
No color.
So
Okay,
if
sir.
there is no color, it is negative. If there is little bit of color, infection is there, but we can measure the load of the bacteria, virus, or fungus. And if it is dark color, then again the load is very high. So
Okay, sir.
this way the results are calculated. Okay. Now
Okay,
the
sir.
advantage of ELISA as compared to RIA, because both techniques are used. Now the advantage of ELISA is that the reagents are relatively cheap and have a long shelf life. Okay.
Okay,
And
sir.
it is specific, and also it gives good sensitivity and no radiation hazard, because one problem with RIA is that we are using a radiation. So if
Yes,
we
sir.
are using radiation, it is hazardous. Okay.
Okay,
Then
sir.
easy to perform and quick procedures. Equipments are widely available, and it can be used for wide variety of infections. Okay. And it can be used for most type of biological samples like serum, urine, cell extracts, whatever it is. So these are the
Okay, sir.
advantages. Now disadvantages that like, because we are measuring the activity of an enzyme, so sometimes it can maybe more complex than the measurement of activity of some time of radioisotopes. Then enzyme activity may be affected by the plasma constituents, right? So sometimes what
Mm.
happen, like you see that if we did not separate the serum properly,
Mm.
then we will get the color in the sample. If the sample
Yes,
is
sir.
colored, then it will interfere with the analysis, right?
Okay, sir. Okay, sir.
Then sometimes the kits are costly, but yes, mostly it is very cost effective. Now it is very specific to a particular antigen, but won't recognize other antigen. That means it is highly specific. Okay.
Mm.
Then sometime, like if there are mutations, then we may have false positive or false negative results.
Good. Yes, sir.
So these are some of the limitations of the ELISA and application. I think you can go through the slides, and if in case if you have any query, we'll be happy to discuss. Okay. Now let me share with the second presentation. Yes, Samra, can you see it? Yes, Samra, are you there? Yes, Samra. Yes, Samra, can you hear me? Yes, Samra. Yes, Samra, can you hear me?
Yes, sir.
I think your connection was lost, eh?
Yes, sir.
Okay, then. Now we will discuss about the second technique, that is radio immunoassay, and it is short form. It is known as RIA, RIA. Okay,
Ria.
and it is
Yes, sir.
same like ELISA. The only difference is that we have changed the this thing. We have changed the analyte. Like we have changed the method of detection. So there, the method of
Hmm.
detection was based on a spectrophotometer, which detect color. Now you know that like if I ask you a like a spectrophotometer, which is based on like two kind of spectrophotometry you might have I think heard about. One is called absorption spectrophotometry, another is called emission spectrophotometry. So in ELISA, the detection is based on absorption of color. Okay, absorption of light by color. Okay, so it is absorption spectroscopy.
Yes, sir.
Now, what is the sensitivity range of absorption spectroscopy? Any idea?
No, sir.
Okay, means the concentration of the color compound produced, it should be in micromolar concentration. It should be in micromolar
Okay, sir.
concentration. Are you
Micromolar
getting it?
concentration.
It should be in micromolar concentration, right?
Yes, sir.
But you know, like most of our hormones, they are in say nanomole or picomole concentration. So sometimes it becomes very difficult to analyze a molecule from ELISA. So instead of using ELISA, we prefer to use RIA. We prefer to use RIA. Okay. So I was telling you that
Yeah.
absorption spectroscopy we generally use. There is another kind of spectroscopy which is having high sensitivity as compared to say absorption spectroscopy. So UV visible spectroscopy is absorption. There is another type. You must have heard about fluorescence. So fluorescence gives us much more or better sensitivity as compared to UV visible spectroscopy. So in UV visible spectrophotometer, we can measure the concentration of an analyte at in the range of say micromole, whereas in ELISA in fluorescence we can measure up to say nanomoles, easily up to nanomoles. But if we have to go beyond nanomole, then we use RIA. Then we use RIA. Can you hear me, Samra? Yes, Samra, can you hear me?
Hello, sir. Can you hear me?
Yes, I can hear you, but you are lost in between. Hello.
Hello.
I think you are getting phone and you are
Hello.
using
Yes,
your
sir. Now
phone.
I can hear you.
Okay. So
No, sir.
if
Not at all.
if we need more sensitivity, then we prefer RIA. Okay. So like simply you
Okay.
compare the
Okay, sir.
ELISA with RIA. I'll not go to history. I'll just go to one picture so that then we'll discuss it. Okay. So this is the picture here. So
Okay,
what
sir.
happens actually that in the in the plate we already have a antibody which is linked with an antigen, and that antigen is having radioactivity, means it is a radio antigen, right? A molecule, a
Okay,
ligand
sir.
which is possessing say radioactivity. Okay.
Okay, sir.
So this
Okay, sir.
radioactivity molecule, it will be generating a stable current, right? It will be, suppose it is a
Mm-hmm.
gamma emitter, or it is a, mostly we use beta emitters. So a like beta radiation is given. That radiation goes to a PMT tube, and from there we get generation of a current,
Current.
like what we have seen in the scintillation counter. So it's a kind of
Yes,
scintillation
sir.
counter, right?
Right, sir.
Now, since if we have a like antibody linked with its antigen, then we are getting a stable current, right? Now,
Okay, sir.
suppose if I put any body fluid here, then that body fluid will be having antigen.
Okay, sir.
Now, that antigen is having radioactivity or it is not having radioactivity?
Sir, it will not have any radioactivity.
It will not have any radioactivity because this we isolated from the sample. It is from the
Sample.
sample. So
Yes, sir.
sample will not have any radioactivity.
Yes, sir.
But this antigen will compete with the labeled antigen with its antibody, because
Okay,
antibody
sir.
doesn't know that which one is labeled antigen and which one is
Which one?
unlabeled antigen.
Unlabeled. Yes, sir.
So labeled antigens means antigen labeled with a
Radioactive,
radioactivity.
sir. Yes, sir.
Okay. So when we mix a sample, then what will happen? The antigen from this sample, they will compete with this antibody, and some of the labeled antigen will be removed because it will compete. Some of the labeled
Removed.
antigen will be removed. So
Yes, sir.
when this labeled antigen will be
Removed.
removed, then there will be decrease in the what?
Current.
Radioactivity.
Current.
And directly decrease in current. So the
Current.
decrease in
Yes, sir.
current is directly proportional to the
Increase in the antibodies of the sample.
no, no, antibody ki to we are not talking about antibody here. That's
No,
why
sir.
I
So
just
sorry. The
that's why I
antigens.
just that's why I brought
Hmm.
you directly on this picture to
Hmm.
understand.
Hmm.
So if the amount of current drop is directly proportional to the amount of antigen present in the sample.
Antigens. Sample.
So
Yes,
higher
sir.
the antigen, more labeled antigen will be displaced and will get lesser current, right? Lower the
Okay, sir.
antigen,
Okay, sir.
lower antigen, labeled antigen will be displaced, and we will get the
Hmm.
high current, right?
High
So
current.
you
Yes,
see
sir.
one advantage is there, that since in ELISA the measurement was based on color, and we need to have an analyte in micromolar concentration. So to get an antigen or antibody in micromolar concentration, there should be significant infection, or the like change in the hormone should be significant. And
Okay,
you
sir.
know, I was telling you that some of the growth hormones in our body, they are present in say picomole concentration or femtomole concentration.
Yes, sir.
And suppose the change in concentration means reduction in concentration from picomole to femtomole will lead to abnormalities, right?
Hmm. Yes, sir.
But measurement from picomole to femtomole itself is a very challenging task.
Yes, sir.
So with ELISA, practically, we can measure up to femtomole. Even if few antigens are present, those few antigens may be like maybe detected. That means any kind of infection can be detected at a very early stage. And second thing
Mm-hmm.
is that any change in the hormonal level can be detected at a very early stage.
Very okay. Okay, sir.
So this is one advantage of
Ria.
RIA to ELISA. Rest is same.
Mm-hmm.
So advantage is
Mm-hmm.
its sensitivity is very high, right?
Mm.
Its
Okay. Okay, sir.
sensitivity is very high. So using antibodies of high affinity, it is possible to detect few pictogram. Picto it is written, originally it
Mm-hmm.
is written as picograms of antigen in the
Mm-hmm.
tube. The
Tube.
greater the
Okay, sir.
specificity of the antiserum, the greater the specificity of the assay. This is
I
the
see.
major advantage. But some disadvantage are associated with that. That use of radioactivity hazardous. Like you
Mm-hmm.
cannot use RIA in routine laboratory setup.
Mm. Okay, sir.
ELISA, you will find you go to a small diagnostic lab in any city. So in a
Mm-hmm.
small room, they are keeping ELISA and they are doing
Eliza.
it.
Yes, sir.
But like RIA cannot be done like this because we need to have a sophisticated laboratory which is
Mm.
having radiation protection capability to run ELISA, run RIA.
Mm.
Okay, that is
Okay. Okay,
one
sir.
major problem that we face with RIA. Then the equipment is also very costly as compared to
Eliza.
ELISA because we need a gamma or a beta counter.
Mm-hmm.
And then like mostly like iodine we are using, it generate gamma radiation, and we know that gamma radiation is highly mutagenic. So we need to
Mm.
have a very protective setup.
Yes, sir.
Then like the body concentrate iodine atoms radioactive or not in the thyroid gland, which are not incorporated in thyroxine.
Mm.
So
Thyroid.
sometimes like
Yes, sir.
some of the body atom may interfere with the analysis of
Mm.
the in
Ria.
this particular.
Mm.
Okay,
Okay,
so
sir.
I think you understood both the techniques well.
Yes, sir.
And with that I have finished up my classes. And
Yes,
I
sir.
think I have shared with you all the notes
Yes, sir.
that will be helpful for you to read.
Mm. Yes, sir.
And you read your notes, and in case if there is any problem, because
Mm.
now today onwards I may not be officially I may not be interacting,
Mm.
but
Yes, sir.
yes, I am available on phone. So whenever you
Okay, sir.
feel like that you want to ask me anything, just leave me a message, and then I will
Okay, sir.
respond to the message and we can discuss it.
Okay, sir. Okay, sir.
Okay. So hope my
Okay.
discussion with you was fruitful and you were able
Yes,
to
sir.
understand something. Yeah?
Yes, sir.
Okay then. So wish you
Okay,
all the
sir.
best and
Thank you, sir.
keep in touch.
Okay, sir. Thank you, sir.
Okay then. Bye bye.